involved. Okay, actively involved. Next, we will discuss one by one. Uh, Coming to the first hypersensitivity reaction. Okay, first hypersensitivity reaction, we can call it as immunoglobulin E, immunoglobulin E mediated hypersensitivity. Because in this type of hypersensitivity reaction, immunoglobulin E play a key role. This is one of the class of the immunoglobulin what we have discussed in the earlier classes. Immunoglobulin E play a key role in making a hypersensitivity reaction. If they playing a role, it play a key role in in this type of hypersensitivity. Immunoglobulin E class antibody binds with high affinity to FC receptors on the surface of tissue mast cell. Okay, and blood basophils. Okay, these two types of cells are having FC receptors. Okay, FC region receptors for immunoglobulin E antibodies. Okay, whenever these immunoglobulin E antibodies get attached with the antigen, and these uh, immunoglobulin FC receptors on the mast and basophils, whenever these cells are get sensitized by the antibodies, and in the second subsequent reaction, subsequent infection can be uh, activate the degranulation process. First, before going to detail the functions, what are the functions of these basophils and mast cells, we will discuss. Mast basophils produce effector molecules capable of ingesting foreign particles and association with parasite infection. Whenever we get a parasite infection, these cells will get activated and fight against those parasite uh, elements, etc., etc. And coming to the mast cell, and these cells releases the granules containing histamine and other active pharmacologically active ingredients. Whenever these cells get activated after getting the information from the antibodies, the cross linked antibodies, cross attached antibodies with the antigen, this mast cell get activated, and after the uh, activation, the sensitization and degranulation will be taking place after the activation of the mast cell, and in the subsequent or second exposure, these mast cells are get activated. I will tell you what exactly happening in this hypersensitive one reaction, and characteristics of different characteristics and mast cells. Okay, and don't worry about this slide. And this is how the hypersensitive one reaction will taking place. And first, what happening in the first step, when immunoglobulin E are located on the mast cell, they are called as sensitized. Okay, and the allergens. Okay, there are different types of allergens are there. For example, proteins. For example, if you take some proteins, foreign serum, okay, foreign vaccines. Vaccines also sometimes act as a allergen, and plant pollen, ray grass, ragweed, timothy grass, birch tree, different types of tree particles also act as a antigen sometimes, and drugs. Penicillin sometimes act as a antigen. Sulfanilamide, local anesthetic, salicylates. These are the drugs. Sometimes acts as a allergen. Okay, and foods also, sometimes food particles also acts as a antigens. For example, nuts, okay, seafoods, eggs, peas, beans, milk. Sometimes these foods, if those people are having allergy reaction, if you take if these foods are ingested by those persons, they will get allergy reaction. And insect products, some insect products also acts as a antigen, bee venom, wasp venom, ant venom, uh, cockroach calyx, and mites. The dust mites also sometimes acts as a antigen. So different types of antigens are there. Uh, for those antigens, whenever the antigen interacted with the antigen presenting cell, these antigen presenting cell sometimes B lymphocyte, macrophage can also act as the antigen presenting cell, dendritic cell in place of B cell. Different types of antigen presenting cells present the antigen to TH cell, okay, TH cell. And whenever the antigen presented by the antigen presenting cell to the TH cell, and these TH cell get activated and proliferated into memory cell and effector cell. That is one story. And in this particular hypersensitivity reaction, what happens that B cell play a key role, because these all the three types of the hypersensitivity reactions are uh, antibody based. So sensitized B cell, whenever the antigen contact with the B cell, and this cell gets sensitized with the help of TH cell, and after sensitization, the B cell proliferated into memory cell and plasma cell. This is a common phenomena. And plasma cell is a specialized cell and specific to the synthesizing the antibodies, and these antibodies are class switched with the specific antigen. Okay, and especially in this type 1 hypersensitivity reaction, immunoglobulin E is a releasing antibody, synthesizing antibody in all cases, hypersensitive 1 reaction. So these immunoglobulin E antibodies goes and binds with the mast cell and basophils, and which contains FC receptor for the uh, immunoglobulin E antibodies. And in the first reaction, this is the first exposure. And this step also called as sensitization phase. Okay, this is the sensitization phase. What happens in the sensitization phase? Class switching of antigen specific B cells will be released. Antibodies, that is specific immunoglobulin E antibodies, and these antibodies goes and binds with the different types of, especially two cells. Okay, there is mast cell and basophil. Okay, and whenever the antibody E goes and binds with the mast cell or basophil cell, and these mast and basophil cells get sensitized in the first step. Get sensitized in the first step. No allergic reaction will be happen in the first exposure, but subsequent exposure, whenever we get uh, same allergen get encountered by the same allergen, and this sensitized mast cell directly recognizes the antigen, that is allergen, by the help of antibodies, with the help of antibodies which those are attached to the FC receptor. And this immunoglobulin E antibody, the paratope, especially paratopes, attached to the epitope region of the antigen. And in this attachment, this binding facilitates the uh, cross link of the antigen. Okay, cross link of the antibodies, cross link of the antibodies. Okay, and a later exposure to the same allergen cross link the membrane bound immunoglobulin E on the sensitized mast cell or basophil, causing degranulation of the cell. Okay, this cross-link antigen, this cross-link antibodies with the antigen provoke the stimulate the degranulation process by the uh, signaling mechanism. It in the uh, plasma with connection to the plasma membrane by the different types of the signaling mechanisms will be activated, and the degranulation process will be as the subsequent consequences of the allergen attachment or cross-linked membrane bound immunoglobulin G. And what happens in the after the degranulation? Pharmacologically active mediators uh, will be released into the different sites. For example, smooth muscle cells, and it reaches the smooth muscle cells and smooth blood vessels, mucous gland, blood platelets, sensory nerve endings, and eosinophils. Okay, and different eosinophils. These different types of the uh, areas will be the target for these pharmacologically active ingredients. Okay, what happens after the getting signal from the these uh, active ingredients? These active ingredients can be categorized into two types. Okay, the degranulation, after the degranulation, active pharmacologically ingredients will be released, and these active ingredients can be categorized into two classes. Okay, that is primary effector mediators. Okay, secondary effector mediators. I will show the diagram. This is the initiation of degranulation. I will explain once again. Uh, okay, allergen cross-linked of cell-bound immunoglobulin E. This is the allergen, and these are the uh, this is allergen. 1 minute. Okay, this is allergen, and these are the immunoglobulin E molecules. And whenever these type of cross linkage of cell bound immunoglobulin E uh, is taking place, this cross linkage give a signal cascade, activates the signal cascade in the cell, that is the mast, especially mast cell and basophil cell, and the result is the degranulation. Now, finally, degranulation process is enhanced by the enhanced of the calcium influx by the ionophore that increase the membrane permeability. For your reference, for your understanding, I will show one another image for you. This is how the degranulation process will be taking place. It's a simple uh, signaling mechanism to activate the degranulation process. What happens whenever the allergen contact
Excuse me,
get
sir.
contact with the uh,
Sir, can you make the screen big because it is not visible? I'm
uh,
not able to
can I
understand.
can I
PPT
zoom?
size. Yeah, yeah.
Okay. Okay.
Right, sir. Thank you.
Uh, what happens in the first step? In the first step, this allergen reaction, this allergen goes and binds with the immunoglobulin E uh, anti-paratope, especially, and this whenever this cross linkage is there, and the inner membrane reaction, that is especially this is one of the cascade system. If you have an idea about cell signaling process, okay, this is all the cell signaling process. uh, IP IP to don't bother about the definition, just this is not a great matter to you, and this is not for you, but I am explaining about this one. And uh, after these 4 5 steps, what happens is this endoplasmic reticulum get activated by the IP3 molecule. Okay, and it releases the calcium ion. Okay, calcium goes and binds with the granules, and this granule, whenever this calcium goes and binds with the granules of the mast cell, and this granule goes and binds with the plasma membrane, and after the microtubule and microfilaments constriction, the degranulation process will be uh, taking place. Okay, that is not a important for you. Just I have shown you the information. Don't bother about this signaling pathway. Just remember the
Okay, sir.
hypersensitive reaction one. Okay, what happens in the hypersensitive reaction one? First step, in the first step, sensitization. Sensitization. Write down about the sensitization phase. Okay, this is the
Okay, sir.
first step in the phase one. First step in the hypersensitive reaction one, and the second phase is that effector phase. After the sensitization, subsequent contact by the same allergen, what happens? This sensitized mast cell, having which is uh, bound with the antibodies, especially immunoglobulin E antibodies, goes and directly recognizes the antigen. Okay, and in this process, the cross-linked antibodies will be the one of the play a key role, and this cross-linked antibodies stimulates the degranulation process or initiate the degranulation cascadial mechanism. After the initiation, the degranulation will be taking place by the cascadial different types of different steps of the cascadial reaction. Because in this process, calcium play a key role. Okay, calcium play a key role, and after the degranulation, two types of effector mediators. So that is, those are the pharmacologically effector mediators will be released from the mast cell or basophil cell. For example, uh, I will show these are the mediators. Okay, upper one, those are the primary. Okay, in this slide, they have given primary effectors. These are the primary, and these are the secondary effectors. So coming to the primary effectors, for example, histamine, heparin, serotonin, okay, neutrophil chemotactic factor, protease. Okay, these are the specialized primary effector mediators released from the activated mast cell and or basophil cells. And what are the consequences? Whenever the histamine and heparin released from the basophils reach to the different tissues, it increases the vascular permeability and smooth muscle contraction. Okay, this is the result of the histamine and heparin. And whenever the serotonin released from the mast cell, it reaches the different types of tissues and increases the vascular permeability, smooth muscle contraction, same, and neutrophil chemotactic factor attracts the neutrophil uh, to the site, okay, site, and whenever the eosinophil chemotactic factor released from the mast cell, it is one of the primary effector mediator. It attracts the eosinophil, okay, to the site, to the infection site, and proteases, okay, also the primary mediator, and bronchial mucus secretions will be the result after the reaching the proteases to the lungs region. Okay, and degranulation of blood vessel basement, degradation, not degranulation, degradation of blood vessel basement membrane, and generation of complement split products, generation, not degeneration, generation of complement split products. And all these reactions uh, may lead to the deleterious effect of on the various sites. Okay, and if we come to the secondary uh, effector mediators, those are the especially leukotriene, prostaglandin, okay, platelet activating factor, and this may increase the platelet aggregation at the site, and degranulation also, and contraction of pulmonary smooth muscle. Okay, this is a very most and dangerous scenario, which may lead to sometime lungs arrest. Okay, and leukotrienes increase the vascular permeability, contraction of pulmonary smooth muscle. This is also one of the lungs arresting factors. And prostaglandins, vasodilation, contraction of pulmonary smooth muscle, platelet aggregation at the site. Bradykinin, increase the vascular permeability, smooth muscle contraction, and all these factors uh, damages the uh, different various whenever these factors reaches to the different sites. Example, for example, what are the sites? Especially these are the sites. What are they? Smooth muscle, smooth muscle, and small blood vessels, mucus glands, blood platelets. Okay, sensory nerve endings. Eosinophils are the target, main target, and these whenever this pharmacological ingredient goes and binds with them, the subsequent reactions, different types of uh, reactions will be taking place, increasing the vascular permeability, increasing the platelet aggregation, different types of reactions. Okay, and this is how uh, the type 1 hypersensitivity reaction affects the tissue. Okay, we can call it as anaphylaxis. Okay, anaphylactic reaction, anaphylactic shock. Sometimes this may lead to the death. What is anaphylaxis? Phylaxis means protection. Okay, phylaxis, meaning of the phylaxis terminology, the word the is protection. Anaphylaxis means opposite to the protection. Protection. Okay. Exposure to antigen can result in the potentially lethal sensitivity to the antigen of the exposure. Whenever this second time exposure is taking place, then the subsequent reactions may damages the tissue site. Okay, may damage the organ, may damage the different tissue sites. Okay, this is anaphylactic shock or anaphylaxis. And uh, examples, different types of examples are there. Okay, diseases, diseases are there. These are the common allergens associated with type 1 hypersensitivity. We have discussed in earlier. Okay, once again I will tell you for you. Proteins, foreign serum vaccine, plant pollens, different types of plant pollens also make a allergic reaction in different types of persons. Okay, those are sensitive to this allergens only. Drugs, some people may get allergic reaction to the penicillin also. Okay, sulfanilamide, different types of drugs. Sometimes foods also act as antigen. Some people in some people milk also cannot digest it after the ingestion. Okay, and especially milk also acts as a antigen in those type of particular peoples. Insect products, especially bee venom, wasp venom, ant venom, cockroach calyx, and different types of dust mites can act as a antigen. And these anaphylactic reactions means uh, making a adverse effect on tissue site. Okay, anaphylaxis means against the opposite to the protection. Okay, whenever the second exposure is taking place, those uh, fighting site. Okay, wherever the immunological reactions are taking place at that site will be damaged. Some type, some tissue can be damaged in that type of reaction. That is anaphylaxis. We can classify into these anaphylactic reactions into two types. Systemic anaphylaxis, one is systemic anaphylaxis reaction. Another one is local anaphylaxis reaction. Local and systemic. Okay. What is systemic anaphylaxis reaction? Whenever we uh, uh, bitten by the bee venom, okay, wasp, okay, hornets ant stings, these venoms act as a uh, antigens and get hypersensitive on reaction. And this is not a particular site. Total system can be damaged. Sometimes may leads to the death. This type of the anaphylactic reactions are called as systemic. No single organ will be cannot be damaged in this type. To entire system can be damaged in this type of the systemic anaphylaxis reaction. But what happens in the localized? In localized means specific regions, specific organs or regions can be involved or damaged in this type of reaction. For example, reaction is limited. Reaction is limited to the specific target tissue or organ in this type of localized anaphylactic reaction. But entire body can be uh, uh, affected by the anaphylactic reaction in the systemic anaphylactic reaction. What happens in the localized reaction is limited to specific region or tissue. For example, allergic rhinitis, asthma, allergic rhinitis, wherever we get uh, infection, those sites uh, will be damaged. For example, I will show this is urticaria. Urticaria also one of the localized infection or anaphylactic reaction. And asthma. Asthma also one of the example for the localized anaphylactic reaction in which our respiratory organs will be the target. Okay, our respiratory organs. Okay, and food allergens sometimes these are the different types of the localized reactions, anaphylactic reactions.
What is the treatment
And
for this?
I am coming to that. I am coming to that point. Okay, these are the atopic eczema, another example, and different types of treatment methodologies are there. And by using drugs, okay, by using drugs, we can control this type of hypersensitivity reaction. One, especially antihistamine drugs. Okay, antihistamine, and in this drug goes and binds with the histamine, release the histamine. Okay, and this histamine, whenever the histamine released from the mast cell or basophil, and histamine can be blocked. Histamine receptor, histamine total histamine compound can be blocked in this by the drugs. Those drugs are called antihistamine drugs. And chromoline sodium can be given for the hypersensitive one reaction to treat the hypersensitive reaction one. And another drug is that theophylline. Theophylline also one of the another drug to treat the hypersensitive one reaction. Epinephrine also give as a drug in the hypersensitive reaction for the hypersensitive reaction one. Cortisone. Cortisone also acts as a drug to treat the hypersensitive one reaction. To uh, mitigate the problem in the hypersensitive reaction one. Okay, these are the drugs. Whenever the person suffering from the hypersensitive reaction one, the doctors will recognize the problem and gives these drugs. Okay, what they do? For example, epinephrine is a drug of choice of a systemic anaphylactic reaction. Okay, person suffering from the systemic anaphylactic reaction, in those persons, for those person, epinephrine acts as a excellent drug. What they do, what it do, it relaxes the smooth muscle. Okay, what histamine can do, it contracts the after the reaching the histamine to the muscle cell, it contracts the muscle, it contracts the everything. And in that type of reaction, after the giving the epinephrine goes and binds with the muscle cells, relaxes the smooth muscle. It relaxes the smooth muscles and reduces the vascular permeability. Okay, vascular permeability to the different types of uh, cells can be reduces. Okay, can be reduces, and improves the cardiac output. After the in the hypersensitive reaction one, cardiac output can be reduced after getting the signal from the histamine, serotonin, bradykinin. It will reach whenever these products, pharmacologically active ingredient, reaches to the myocardium muscle. Okay, cardiac muscle, and these muscles can be uh, cardiac output can be uh, limited, okay, decreased after reaching this histamine, serotonin, bradykinin to the myocardic muscles. And by giving the epinephrine, we can improve the cardiac output also. It increases the cyclic AMP levels in the cells. Okay, after giving the epinephrine, it increases the cyclic AMP level. It blocks the degranulation. Okay, cyclic AMP is a one of the components. I will show the role of the cyclic AMP. Don't bother about the role and mechanisms. Just remember the things. What is the hypersensitive reaction? What are the steps? Enough. Okay. In this mechanism, in this mechanism, cyclic AMP will be released. Okay, and goes and binds with the granules. Okay, and it blocks the releasing of the calcium ions from the endoplasmic reticulum by the different by activating the different cascadal reaction. And whenever the calcium is not available to the not available, the degranulation process will be blocked. And by this mechanism, epinephrine can also act as a one of the wonderful drug for the treating the hypersensitivity reaction. One, this is the medical uh, information. So, any doubts on hypersensitivity reaction one?
No, sir.
Okay. Coming to the hypersensitivity reaction two. Okay, hypersensitivity reaction two, also known as antibody-mediated cytotoxicity. Antibody-mediated cytotoxicity. What we have discussed in the uh, transplantation reaction, okay, transplantation rejection. Okay, this type of hypersensitivity also play a key role in the transplantation rejection. And 2 and 4 hypersensitivity reaction play a major role in the transplantation rejection reactions. Okay, graft rejection. Okay, we can also know also known as antibody mediated cytotoxicity. Okay, antibody mediated cellular antibody mediated cell mediated cytotoxicity, ADCC, antibody mediated cell mediated cytotoxicity. Okay, and what happens in this uh, hypersensitivity reaction 2? In this hypersensitivity reaction 2, immunoglobulin G play a major role. Okay, immunoglobulin G and sometimes immunoglobulin M also play a major role in this type of reaction. Okay, antibodies directed against the cell surface antigen. Okay, mediates the cell destruction via complement activation or antibody dependent cell mediated cytotoxicity. Whenever our body get encountered by the different types of pathogen or parasite or different types of antigens. Okay, and what we have talked earlier. Okay, and for these antigens, specialized antibodies will be produced. Okay, by the activation or by the activation of the B cell from the plasma cell. Antibodies are produced. Those are the immunoglobulin M and immunoglobulin G. After production of the immunoglobulin M and immunoglobulin G, and these antibodies goes and binds with the different types of cells, immune cells. For example, uh, different types of cells in the sense monocytes are having FC receptors, same like our mast cells. Mast cells are having FC receptors for immunoglobulin E. Okay, in this type of reaction, monocytes monocytes are having FC receptors for specialized antibodies. Those are immunoglobulin G, immunoglobulin M, and neutrophils also are having FC receptors for these two immunoglobulins, and eosinophils are also having FC receptors for the immunoglobulin G and M, natural killer cells. Natural killer cells also having this type of receptor. What happens whenever the immunoglobulin G and immunoglobulin M uh, produced from the plasma cell, this goes and binds with the different types of cells. Those are having receptors for this antibody. Monocytes, neutrophils, eosinophils, natural killer cells. What happens after the uh, uh, attachment to the Fc receptor by the uh, these antibodies? Whenever the antibodies are attached to the monocyte, phagocytosis will be enhanced after the attachment. And whenever these antibodies goes and binds with the neutrophils, okay, destruction lytic enzymes will be released after the attachment of the antibodies to the neutrophils, eosinophils. Okay, degranulation will be the result after the attachment to the uh, this eosinophils. Natural killer cell, whenever these immunoglobulin G and M goes and binds with the natural killer cells, this also may a natural killer cell get activated after the attachment and kills this site, kills this cell, target cell or target tissue. And this is the antibody dependent cell mediated cytotoxicity. These are the cells: monocytes, neutrophils, eosinophils, natural killer cells are the cells. These cells are getting information, are getting activated after the get activated after the attachment of the uh, immune antibodies. And what is the consequence? The target tissue will be damaged after the attachment of the immunoglobulin G and M to these cells. And sometimes what happens? Sometimes opsonization and complement uh, protein, these whenever the immunoglobulin G is there, and immunoglobulin G also having the complement protein receptor. Okay, this complement protein goes and binds with the immunoglobulin G, and what is the consequence? The target tissue will be damaged by the activating the complement system, making a pore on the cell on the surface of the target tissue. These whenever these antibodies goes and binds with the target tissue, and these complement proteins also binds with the antibodies. Okay, and this binding can facilitate the complement activation and by making a force on the target site. These are the two. By these two ways, hypersensitivity reaction may lead to the hazardous effect on affected tissue, infected tissue. Okay, for example, our blood transfusion reactions. Okay, sometimes those persons getting the blood from the uh, donor, if the donor and recipient blood groups will be different, okay, what is the consequence? The consequence is a agglutination, okay, destruction of the entire system. By this hypersensitive reaction too is involving in those type of reactions, blood transfusion reaction. If the man if their blood is not matched properly, hypersensitive to reaction will be the result. Are you following me?
Yes, sir.
Yes, sir.
Another example is that erythroblastosis fetalis. Did you hear this uh, sentence? Erythroblastosis
Yes, sir.
phenomena.
Pregnancy
Yes, sir.
time.
Fetalis.
Yes, sir.
Second
Okay.
baby.
In the in this process, second baby will be the target. What happens in the first delivery? In the first delivery, antibodies will be produced to the baby's blood. That is Rh positive. In the subsequent delivery, already antibodies are there in the mother's body. And what is the consequence? For the second baby, uh, these antibodies cross the placenta. Okay, especially antibodies G, okay, blood group antibodies are G. These formed antibodies entered into the baby's blood circulatory system. Antibodies and get attached with the blood group, and it damages entire blood circulatory system or blood cells. Okay, it damages the blood cells by the activating different immune processes. Okay, this is one of the another example for the hypersensitivity reaction too. Okay, you know about these entire things. Okay, Rogan, Rogam is a drug. Okay, Rogam is a drug. It is a antibody to the uh, Rh positive antibody. Okay, are you understand? Rogam is an antibody.
Yes, sir.
It
Yes,
prevents the
sir.
B cell activation. It prevents the B cell activation and memory cell formation. It prevents the B cell activation. Okay, memory B cell will be there in the mother body, and those memory B cell will be blocked after the giving the Rogam. Rogam is a drug name, and this is one of the treatment for this erythroblastosis fetalis. And also different types of autoimmune hemolytic anemia reactions also example for this uh, second type of hypersensitive reaction. Especially blood group antigens uh, make play a key role in this type of reaction. Okay, complement system also involved in this type of reaction. Okay, and different types of cells, um, macrophages. Okay, neutrophils, eosinophils, natural killer cells also actively involved. That's why name has given cell mediated, antibody dependent cell mediated cytotoxicity. Okay, these cells are get activated after the attachment of the by the immunoglobulin G, immunoglobulin M, and after attachment, monocytes, neutrophils, eosinophils, natural killer cells get activated. It damages the target cell, the target cell. In this process, the hazardous, deleterious effect on the site is a main consequence. Okay, this is about a hypersensitive reaction too. Okay, is it clear?
Yes, sir.
Okay.
Yes, sir.
And coming to the hypersensitive reaction 3. Hypersensitivity reaction 3, also known as immune complex mediated. Okay, in this hypersensitivity reaction, antibody also play a key role, and these three types of hypersensitivity reaction. But antibodies are different. Okay, first reaction immunoglobulin E play a key role. In the second hypersensitivity reaction, immunoglobulin M and G. Third reaction, immunoglobulin G, immunoglobulin G, whether the immunoglobulin G or any immunoglobulin, and wherever the complex is important, whether they belong to the class A antibody, G class antibody, M class antibody, etc., etc. Wherever the antigen antibody complexes are there, our body especially removes clears the antigen antibody complexes. You know about these things. Uh, in that process, sometimes what happens in that process, the target tissue, the deposited tissue also can be damaged in the removing process, in the clearance process. What happens in this situation? Antigen antibody complexes deposited in various tissues, for example, kidney, uh, and brain, sometimes meninges, layers, sometimes different uh, areas, etc., etc. Okay, what happens? It tissues induces the complement activation. Okay, whenever the you have studied about the complement system, classical pathway, and alternate pathway, what happens in those pathways? Complement classical pathway complex immune complex play a key role to activate the uh, classical pathway. Antigen antibody complex play a key role. Whenever the antigen antibody complex is there, complement protein goes and binds with the antibodies. All antibodies are having especially immunoglobulin M and G, are having receptor for the complement proteins, and these complement proteins, and especially C3b goes and binds with the antibodies. What happens? It activates the complement activation, mast cell degranulation, okay, infiltration of neutrophils to the site, and different mechanisms get activated after the formation of the uh, complex, antigen antibody complex. Wherever the antigen antibody complexes are formed and deposited, and this is the consequence. Okay, what happens? And a complement protein goes and binds with formed antibody antigen complex, and after the contact with the antibody uh, by the complement protein, it get activate the subsequent cascadal reaction. One by one, all the complement proteins goes and binds with the complement proteins subunits, and it makes a pore on the target site or tissue, uh, especially. Excuse me, membrane attack complex. Okay, and mast cell degranulation is one of the subsequent reaction. If it is immunoglobulin E, okay, if it is the antigen antibody complex is the immunoglobulin E, mast cell gets sensitized and destruct the entire affected tissue. Okay, and infiltration of neutrophils is one of the chemotactic and opsonization signal by after the attachment of the complement protein. These are the consequences after the attachment of the complement protein to the antigen antibody complex. Okay, these are the consequences. Complement activation is one of the consequence. Mast cell degranulation, another consequence. Infiltration of neutrophils is one of another consequence. All these consequences may lead to the deleterious effect on target tissue. And what happens after the neutrophil infiltration to the site? Neutrophil releases the lytic enzymes and kills the target tissue or cell. Okay, and phagocyte phagocytosis will be one of the another consequences after the coating by the C3b to the antigen antibody complex. Okay, phagocytosis, neutrophil target lysis, and complement activation, mast cell degranulation. All these pathways uh, destruct the infection site. Okay, for example, uh, Arthur's reactions. All Arthur's reactions are the hypersensitivity 3 reactions, and serum sickness also one of the example for the hypersensitivity 3 reactions. Necrotizing vasculitis, glomerular nephritis. Okay, glomerular nephritis is a major consequence of the hypersensitivity 3 reaction. Rheumatoid arthritis. Okay, our synovial fluid will be damaged. Okay, in the knee region, all the antigen antibody complexes will be deposited. Okay, by result of the hypersensitivity reaction, all the synovial fluid will be get damaged after the in the process of uh, removing the antigen antibody complexes from the synovial fluid. What is the consequence? Sometimes entire synovial fluid will be damaged. Okay, in this process, that may lead to the rheumatoid arthritis. The systemic lupus erythematosus is one of the another consequences, which is the autoimmune disorder. It's also autoimmune disorder. We will discuss at that time. The systemic lupus erythematosus. Okay, this is the autoimmune disorder. Okay, you know about all about these things. Okay, antigen antibody complexes. Wherever these antigen antibody complexes uh, is formed, it should be eliminated or it should be cleared from those sites. In that process, our own tissue will be damaged by the different types of immunological active pathways. Okay, active reactions. Okay, anaphylotoxins, lysis, vasoactive amines, different types of mast cell. vasoactive amines. Okay? Different types of pharmacologically active ingredients damages our own cells. If it is our own synovial fluid, that may leads to the rheumatoid arthritis, glomerular area, glomerular nephritis, meningitis. Different types of uh, disorders will become, diseases will become after the came into the picture after the hypersensitivity reaction 3. Is it clear, Amma?
Yes, sir. Sir.
Simply remember that the third type is that antigen antibody complex reaction. Okay? Mediated hypersensitivity. And come to the fourth type of hypersensitivity reaction. Uh, This is fourth type. We can also call it as DTH. What is DTH? Delayed. I have clearly explained this type of hypersensitivity in the
Delayed type
transplantation.
of hypersensitivity.
Delayed type of hypersensitivity reaction. Whenever we have, when I was talking about graft rejection, okay, I have clearly entire process have been clearly discussed at that point. Once again, I will explain one more time. Anyway, and delayed type of hypersensitivity reaction, also known as cell-mediated hypersensitivity reaction. Okay, because in this hypersensitivity reaction, no antibodies are play a key role. Okay, no antibodies are uh, play a part in this making a hypersensitivity reaction 4. Okay, are not in no antibody are not involved in this type of reaction. Only cells, especially T cells, B cells. Okay. Tell me, for example, entire this reaction is cell-mediated immunity. Okay, if you have an idea about cell-mediated immunity under the acquired immunity branch, you get uh, entire knowledge about the fourth reaction. If you have an idea about humoral immunity sub-branch under the acquired immunity, you may get entire information, entire knowledge about the 1, 2, 3 reactions, hypersensitive reactions. Okay, you can understand excellently. This fourth type of reaction, also known as cell-mediated hypersensitivity and sensitized TH cell. This hypersensitivity reaction uh, taking place by the two phases. Okay, one is that sensitization phase, another one is effector phase. One is that sensitization phase, another one is that effector phase. What is sensitized phase? Sensitized phase means those cells. Okay, what we have been talking about T cell and B cell. Those cells get sensitized after taking information from antigen. If you take T cell cannot directly recognizes the antigen. It requires the antigen presenting cell. Then only the antigen can be processed by the T cell. It cannot recognize it directly. Antigen presenting cell will present the antigen to the T cell and make and these two cells. Okay, for example, if the antigen presenting cell having the MSC receptor 1, it will present the antigens to the TC cell. If it is having MSC receptor 2, it presents the antigen to the TH cell. Okay, this is the difference. CD4 and CD8. In this picture, you clearly shown that CD4 means TH cell. CD4 TC cell receptor means TH cell. CD8 means TC cell. This is MSC 2 molecule. Okay, it antigen presenting cell is having MSC molecule 2. This is MSC molecule 2. What is the consequence? Whenever the antigen presenting cell presents the antigen to the TH cell, and this is the one first signal. Okay, the communication between MHC molecule and CD receptor is the first signal, and this communication will sensitizes a pH cell, and further sensitization are enhanced by the secretion uh, secreted by the antigen presenting cell, whether by the TH cell itself. Okay, by taking the signal from the antigen presenting cells or itself taking the signal from its own chemo cytokines, it get further activation or a sensitization will be enhanced after getting the information from the antigen presenting cell or its own chemical molecules, and these cytokines can be interferon gamma, okay, interleukin 2, okay, interleukin 3, CSF, MISF. Don't bother about the full forms. Okay, you can remember cytokines. Its own cytokines or and cytokines of antigen presenting cells. This further uh, taking the information from these cytokines, the further enhancement of the sensitization or activation will be taking place after getting information from these cytokines. Okay, what happens after sensitization? And sensitization may lead to the activation and total activation of the T cell. And what happens after the activation? These T cell get proliferated into two cells, memory cell, effector cell. And effector cell will go and bind with the three cells. Okay, the excellent function of the PH cell is that communicating with three different types of cells. What are they? Macrophage, okay, B cell, second cell, CC cell. This cell can bind with these three types of cell, and by taking the information from these TH cell, those cell will be activated, further enhanced, and macrophage will go and do the phagocytosis. B cell will release the antibodies. T cell can damage the entire area by releasing the different types of granzymes, okay, different types of enzymes, okay, different types of enzymes released by the cytotoxic T cell will damage the cell. It may lead to the death of the cell, may lead to the necrosis of this target site. Okay, and this fourth type of reaction, sensitization, is the first phase. You know about the first phase. What is sensitization? And second phase is the effector phase. You also know about this effector phase also. And what are the consequences? What are the major uh, intracellular pathogens may leads to the sometimes infection or infestation by the different types of the infestation by the different types of the pathogen and parasite may lead to the this fourth type of hypersensitivity. For example, intracellular bacteria, Mycobacterium tuberculosis may leads to the hypersensitivity reaction 4. Virus, Mycobacterium leprae, different types of Brucella arbotus, intracellular fungi, pneumocystitis carniae, candida, different species of fungi also uh, activate the hypersensitivity process and may lead to the different types of damages. Intracellular parasite leishmania, okay, different types of parasites also uh, may lead to the hypersensitivity reaction, infection by the infestation by the different parasites also may lead to the hypersensitivity reaction 4. Uh, okay, sensitization phase. Okay, sensitization phase. What happens? The antigen presenting cells, macrophage, Langerhans cells, sometimes B cells also, and this MSC molecule and CD4 contact and this another receptor communication and cytokines communication. All these communications may involving in the activation of the T cell or sensitization of the T cell. And after getting information from these different three signals, okay, first signal, second signal, third signal, this TH cell get activated or sensitized and proliferated into the two cells, that is effector cell and memory cell. And what happens in the effector phase? This TH cell goes and binds with the different types of cells, maybe macrophage, TC cell, B cell, and in this picture they have shown macrophage reaction. What happens after the attachment? Cytokines will be released after the communication between the macrophage and TH cell. Interleukins, interferons, tumor necrosis factors, different types of active ingredients, cytokines will be released from both cells, TH cell and macrophage cell. What is the consequence? The consequence is the activation of macrophage. So the very dangerous uh, phenomena. After getting information from the different types of communication, signal attachment, receptor attachment, cytokines, macrophage will be activated. And what are the consequences? Phagocytosis. Tell me, Amma. Phagocytosis is one of the major consequences, and sometimes it will release the active ingredients. Okay, those active ingredients also damages the target site. Okay, for example,
Autoimmune disease, sir. Autoimmune disease.
autoimmune diseases we will discuss in the next classes. Okay, autoimmune disease we will discuss later. This is not a autoimmune disease.
Okay.
Third type you can understand as a autoimmune disease. Third type. Okay, third type will be the autoimmune disease. Third type may lead to the autoimmune disease.
Okay.
This is not a autoimmune disease. Contact dermatitis and a graft rejection. Okay, graft rejection is a major example for the type 4 hypersensitivity reaction. We have also discussed this reaction in the transplantation topic. Okay. If you have any doubts, you can ask. Any doubts, Amma?
Sir, what is the function of calcium ions in activating the immune system?
Calcium ions goes and binds with the granules. Function is, I am telling about function. Calcium ion goes and binds with the granules. Okay. And after binding with the calcium ion, granule will be degranulated. Okay, granule will be degranulated after getting information from the calcium.
Okay, sir.
Then only degranulation process will be take place. Okay.
Okay, sir. Thank you.
That is the mechanism. The mechanism is there. You can cross check with the signaling, cell signal mechanism. Okay.
Okay, sir.
In the fertilization, you also it also comes in the fertilization chapter. Okay, developmental biology.
Yes, sir.
Sperm. What is the topic name? That is polyspermy. Okay, polyspermy. Sperm contraction.
Yes, sir. Acrosomal function during
after the acrosomal reaction. Only one sperm can be allowed into the ova after contact with after fertilization. The other sperm contact will be reduced by the formation of cortical reaction.
Yes, sir.
Okay. In that cortical reaction, cortical granules will be degranulated. Okay, contents will go and bind with the different types of zona pellusa region. Okay, zona pellucida region proteins, and further attachment of the sperm will be reduced. Okay, attachment can be reduced after degranulation process. Okay,
Yes.
at that time also you have studied that calcium, role of the calcium. Okay?
Yes, sir.
Okay.
Thank you, sir.
Anyone doubts?
So these drugs, when we are giving to the hypersensitive persons, so the once
Mm.
the drugs are given, that will be
Mm.
for that time being only, or for throughout the life they have to take the drugs.
Just ask again.
As you said that there are few drugs which will prevent these hypersensitive reactions. So these
Yeah.
drugs they have to take for throughout the life, or just for
No,
once.
no. Just for once. Just for once. No. We don't want to just take the throughout the life. Just one time.
Okay.
One time. After taking one time, it's enough. No need to take entire lifetime.
Throughout the life. So that leads to the neutralization of that complex.
Just for one reaction. Okay. Whenever we get hypersensitive reaction, at that time those drugs may neutralize the entire pharmacological active ingredient or different types of cell activation. Okay, mast cell activation may be reduced or degranulation. In the sense, degranulation process will be reduced, blocked by taking
Okay.
these drugs. Okay, after taking these drugs goes and binds with the sensitized mast cell and basophil. Whenever these drugs goes and binds with the mast cell and B cell, sensitized B cell, basophil, degranulation means calcium, releasing of the calcium will be blocked from the endoplasmic reticulum.
Okay.
There is no availability of calcium, there is no degranulation.
Okay.
One time is enough. For only one. After few years or few days, whenever we get second hypersensitivity reaction, then we just a second drug also required. Second time also required.
So, for example, in the food allergies, is it okay to take one-time drug and it will function for throughout the life, like memory cell storage?
No. Only one time it's enough. One for hypersensitive one time. Another whenever you get another hypersensitive reaction, it requires another dose.
Okay.
N number.
For example, some people will be allergic to
It is
seafoods,
not.
no, sir.
It is
It is
not
not
a
recommended
vaccine.
to next time eat.
It not communicate next time. Okay, next time we also take one another dose.
Better not
Like
to take,
injection, not
actually.
this is not a like vaccine. It's not actually like a vaccine.
Okay, sir.
Every time we must take
Keep
drugs.
the medicine with us.
Okay. That is
Okay, sir. Thank you.
Next. Tomorrow we will discuss about vaccine or auto immune disorders. Okay, vaccinology or autoimmune disorders. We will discuss about as your wish. So choice is yours. Autoimmune disorder or vaccinology. Unit 13 or unit 10?
Unit 10, sir.
Whatever
We'll go
you-
back,
Unit 10?
Yes, sir.
Okay. I will discuss about unit 10. Okay, autoimmunity and immune diseases. We will
Diseases.
discuss unit 10 as a tomorrow topic. Okay.
Okay, sir.
If you have any doubts,
Thank
you
you,
can
sir.
ask in the next class also. Okay, about
Okay, sir.
hypersensitivity, about transplantation, immunology, you can ask in the next subsequent classes also. Okay,
sir.
Namma?
Okay, sir.
We will discuss autoimmune disorders, okay, diseases. Just tell me about autoimmunity. What is autoimmunity?
Um, our own immune cells will attack our own body cells.
Our own immune cells attack on our
Attack.
own.
Other body cells or organs.
Actually, our immune system recognizes our own antigens as a foreign antigens, and it attacks back to those cells and makes our body organs or tissues damaged.
Yes, exactly. That is autoimmune. Auto means self immune.
Self-recognition power is lost, sir, actually.
Okay. Anyone?
Due to some mutations
Mm.
or else due to aging process.
Okay.
Physical or chemical agents.
Immune tolerance. Immune tolerance, especially in the immunology terminology, we can say it as loss of immune tolerance.
Immune tolerance.
Okay. Immune tolerance.
Yes, sir.
Loss of immune tolerance may lead to the autoimmune disorders or diseases. What is immune tolerance? Our immunity has a six functions. Okay. Our immunity has six functions. It detects the foreign bodies. Okay. Detection of foreign bodies is one of the function, our immune function. And another function is that immune response against the different foreign particles. And third function is that antigenic specificity is one of the functions. And immune memory is one of the function of our acquired immunity. And immunological surveillance is one of the function of our immunity. And sixth function is that immune tolerance. Okay, immune tolerance means self and non-self recognition. Okay, it cannot recognize our own self. Okay, it has an idea about self and non-self. It only recognizes the foreign bodies or foreign cells or foreign pathogens or foreign parasites as a antigen, not our own self. Whenever this knowledge or power will be lost, those in those situations, okay, loss of immune tolerance is a major cause in the autoimmune disorder. Okay, Namu?
Yes, sir.
Loss of
Okay, sir.
immune tolerance. Okay, self recognition, self and non-self recognition will be lost. Okay, by different types of uh, situations, in the different types of situations. Okay, we will discuss what is the definition of the autoimmune disease and what are the types of autoimmune disorders are there and how they are arising or how uh, uh, they are there. They are activating or they are damaging our organs or tissues. We will discuss in the tomorrow class. Okay.
Okay, sir.
Okay.
Thank you.
Thank you, sir.
offline.